Hello, my name is Miguel Ángel Díaz Campos. I'm a Mexican student, and today I will present my work titled Single Cell Analysis to Characterize the Cellular Microenvironment in Human Hepatocellular Carcinoma. So, as an introduction, hepatocellular carcinoma is the most common form of primary liver cancer and remains one of the leading causes of cancer-related mortality worldwide. A key feature of this disease is that its progression and resistance to therapy are not driven by tumor cells alone, but also by the surrounding tumor microenvironment. This includes human cells, stromal cells, and endothelial cells that together shape a supportive ecosystem for tumor growth. Our goal in this study was to use single-cell RNA sequencing data to characterize the composition and functional behavior of the cellular microenvironment in HCC, with particular emphasis on immune suppression, metabolic reprogramming, and intercellular communication. For this, we used publicly available single-cell datasets that include both healthy liver tissue and advanced ACC samples. Our analysis pipeline began with rigorous quality control, including double removal and filtering based on gene count and mitochondrial content. We then performed normalization and dimensionality reduction using PCA and UMAP. For clustering, we applied the latent algorithm, which allowed us to identify transcriptionally distinct cell populations. And to assign biological identities to these clusters, we combined two machine learning-based annotation approaches: CellTypist, which is supervised, and Scanby, a semi-supervised probabilistic model. All labels were manually reviewed and adjusted to improve accuracy. For this, we noted that HCC samples were characterized by a marked expansion of malignant hepatocytes, tumor-associated macrophages, tumor endothelial cells, and myeloid-derived suppressor cells. But to understand the molecular signatures of each population, we performed differential gene expression analysis using HR, comparing the disease samples against healthy controls within each cell type. And it is interesting how in malignant hepatocytes, we observed a strong upregulation of metabolic and mitotic genes such as GCP3 or SIMPA, CCB2, and TOP2A, which point us to a possible enhancement of glycolysis, stemness, and cell proliferation. In other way, the tumor-associated macrophages express markers consistent with an immunosuppressive and an MTLO2-like phenotype. And finally, endothelial cells in tumor overexpression genes involved angiogenesis and hypoxia response. Meanwhile, T and NK cells displayed a significant downregulation of immune checkpoints and antigen presentation genes. And finally, to explore how these cell populations interact, we conduct a cell-to-cell communication analysis using cell phone DB, which infers ligand receptor interaction based on expression data. This allowed us to identify specific communication pathways between malignant cells and the immune or stromal compartments. Notably, we found that malignant hepatocytes signal to endothelial cells and macrophages through ligand receptor pairs, such as transferrin and transferrin receptor 2, C3 to C3RA1, and apolipoproteins to TREM2 receptors. These pathways are associated with iron transport, inflammation, lipid metabolism, and immune suppression. But the most prominent interactions were between malignant cells and TEMs, suggesting a cooperative relationship that it's possibly facilitating immune evasion and metabolic support. With this network of interactions, maybe is highlighting us how tumor cells are not isolated entities, but central orchestrators of microenvironmental remodeling. So, to summarize some of the most important findings, we can say that we identify clear differences in cellular composition and transcriptional state between healthy and tumor tissue. Also, malignant cells display coordinated metabolic and mitotic reprogramming, and immune cells, particularly T and NK cells, are present but suppressed with signatures of exhaustion. Finally, the ligand receptor analysis revealed that malignant cells actively engage with macrophages and endothelial cells to reinforce tumor promoting condition.